So far in the course, we talked about mechanisms involved with the cause of cancer. We discussed some oncogenes that can be the driving force of cancer development, and how tumor suppressor genes, which are protecting us from cancer, can be inactivated. In most cases, we realize that cancer is a result of accumulation of mutations in several oncogenes and tumor suppressor genes. Today, I would like to talk with you about a specific dominant oncogene called the bcr-abl. bcr-abl has a causal relationship with CML, chronic myelogenous leukemia. And it's doing it pretty much on its own as a dominant oncogene. CML is a fatal disease. Without the proper treatment, everybody with CML is likely to die from the disease. In this lecture, I would like to explain the activity of the ABL tyrosine kinase and how the ABL can be involved with CML. And in the following lecture, I will talk with you about a unique treatment that was developed against CML, which is based on this driver mutation. For reading, you can go to Cancer Medicine. Here, chapter 128. It's about chronic myelogenous leukemia, CML. And you can find this textbook in the NCBI website in the bookshelf. Few basic principles of cell division. In general, dividing cells can go towards three different directions. First, self-replication, to make exactly the same cell like the parental cell. Second, cell can differentiate towards mature cells. And the third option is that the cell is going to die by a process called apoptosis. The fate of cells is usually one of the three. In general, any time that we are making a new cell, one cell must die because we need to keep the same number of cells in our body. Cancer is one of those situations that we have more and more cells, mainly because some cells refuse to die or to differentiate. So in general, this is the basic model for the hematopoietic stem cell differentiation. In the bone marrow, we have cells which are very young, but they are already committed to become hematopoietic stem cells, committed to the hematopoietic lineage. Eventually, as mature cells, those cells are going to be found in the circulation. To maintain its own number, the stem cell produces an identical daughter cell. The other daughter cell can enter a phase of proliferation to increase the number of cells and differentiation, ultimately forming many differentiated cells. Stem cell can enter into the proliferation, maturation phase, And by the end of this phase, they reach full maturation towards red blood cells, towards lymphocytes, monocytes, and many others. The cells in the blood circulation are the mature effector cells. Cells such as red blood cells and neutrophils, as you can see them here, are an example of mature cells. Those are the functional cells, and they cannot be divided any longer. After a certain time of doing their job, they enter apoptosis pathway and they are out of the way. Their replacement must be generated from the hematopoietic stem cells. Stem cells can enter the proliferation cell compartment, and eventually they will turn into effector cells. Several mediators and factors can control this process. Among them, factors which are involved with the replication of stem cells, and other factors are involved with the proliferation of cells in the proliferating maturating pool. They can dictate how many cells are going to be made in each compartment. Those factors must be tightly regulated, to be tightly controlled in order to reach the appropriate number of cells in each compartment. Loss of control on this system can lead to problems. If the system is controlled too much and you're not going to have enough effector cells, you're going to have one type of a problem that can be expressed by low level of cells. And here, for example, we can talk about immunosuppression, reduced blood coagulation, etc. So this is one direction. But on the other hand, if those cells are going to be made at higher level from the norm, it can lead to problems. If you have too many cells in stem cell compartment, you are going to add more cells in the proliferating pool and more cells in the effector cells. And in some situations, to have too many effector cells can have a negative effect. Furthermore, too many cells in the stem cell compartment can lead to leukemia. Let's concentrate here about the situation that this system is making too many cells. Malignancy. Malignancy of the hematopoietic cells can arise either in the stem cell compartment or the proliferating compartment. However, in practice, malignancy is more common in the stem cell compartment. And there are a few reasons for this. Stem cells are long-lived, so can potentially accumulate multiple mutations. And by their nature of self-renewal, so the mutation in stem cell is likely to be maintained in the population of stem cells. And if this mutation is making those stem cells to proliferate more, this mutation can take over the cells in the bone marrow. So in general, normally we want to have a certain amount of proliferation in the stem cell compartment. And that's going to lead to certain amount of cells in the proliferating compartment, and we are going to have a certain number of cells as effector cells. However, if the stem cell compartment is going to be overactivated, it can lead towards malignancy. The result is increased stem cell self-renewal, increased survival of proliferating cells. So eventually what you're going to have is that the stem cells are going to make more of themselves. More stem cells lead to more cells in the proliferating pool, and also you're going to have more mature cells as compared to the normal situation. This is what we have at the beginning of chronic myelogenous leukemia. This is the chronic phase of the disease. During the chronic phase of CML, you can see more stem cells, more proliferating pool cells, and more mature cells. The chronic phase can last for several years even without any treatment. Normal stem cells are going to make some stem cells and certain amount of proliferating cells and a certain amount of effector cells. But during the chronic phase of CML, we have more cells in each compartment as compared to the norm. The effector cells function normally. However, genetically, they are different from the norm as they have a certain genetic abnormality. Clinical chronic myelogenous leukemia during the chronic phase. The chronic phase is not normally fatal. Normal bone marrow cells are replaced by CML cells that have a better replication ability as compared to normal cells. Since CML cells mature in great number, excess mature cells are produced. The CML cells are functionally normal. Therefore, anemia, infection, bleeding are usually not detected at this stage. CML cells, however, can accumulate in the blood and secondary hematopoietic organs such as the spleen and the liver, and that can lead to enlarged organs that may cause discomfort or swelling. General tiredness due to increased metabolism to produce the cells can be found from time to time, and those type of symptoms are likely to be the first reason that the patient is coming to see a doctor. Tiredness, or they feel some organs in an abnormal way. Sometimes we can see thrombosis due to excess cells which are blocking vessels. The platelets number is going up as well, and here it can lead towards thrombosis. So the chronic phase seems to be quite benign. This is a phase which is likely to be stable for quite a while. It's very much related to the time the diagnosis took place. Early diagnosis, probably the patient going to stay for longer in the chronic phase from the time of diagnosis. But on average, the chronic phase is between 4 to 6 years. But we know cases that the person stayed in the chronic phase for 20 years from diagnosis even without treatment. But in others, this stage was much shorter than the average. But in all cases, the untreated chronic phase is a time bomb. In all those cases, the patient is going to progress to the accelerated phase, which will follow by blast crisis phase. The acceleration phase is up to 1 year or so before the patient progresses to the blast crisis phase. And this is the terminal phase, which lasts for 3 to 6 months. Everybody in this stage is likely to die from the disease. So chronic phase is a strong indication that the acceleration and the blast crisis is likely to take place. So what are the differences between the chronic phase and the blast crisis? During the chronic phase, there are more cells in each of the compartments, including the effector cells. The differentiation process is working fine. However, it is different when it comes to the blast crisis stage. First, more proliferation in the stem cell compartment as compared to the chronic phase. So you can find more stem cells in the bone marrow and more cells in the proliferating pool. However, those cells in the proliferating pool are losing their ability to differentiate. They are going to stay as young, not fully differentiated cells. They cannot do their job of the effector cells, so they are not functioning well. So beside the fact that we are making a lot of useless cells that can cause damage by themselves, we are not making the effector cells which are supposed to be made from the proliferating pool. Putting both together make the situation to be fatal. CML blast crisis. The abnormal stem cells continue to replicate but undergo with additional mutations. Most of the mutations have no effect, passenger mutations, but in the end a new clone emerges which the ability to differentiate is lost. And this is a result of a driver mutation which can come on top of genetic abnormality that we had earlier. So the relatively benign CML come to resemble an acute leukemia. Here, normal cells are not produced and the undifferentiated blast cells are functionally useless. Therefore, the death is from anemia, infection, or bleeding. So CML is a disease that starts with low level of symptoms and usually without the appropriate treatment is going to move towards the blast crisis stage and the death of the patient. So what is the gene which can be involved with chronic myelogenous leukemia? Earlier we talked about receptor tyrosine kinases, and I mentioned that there are other tyrosine kinases which are not integrated in the plasma membrane, but you can find them in the cytoplasm. The cytoplasmic protein tyrosine kinases are important in many signaling pathways. Many of them are initially discovered as proto-oncogenes of their retroviral transforming protein counterpart, as we discussed about SARC. Almost half of the 32 known cytoplasmic protein tyrosine kinases have been implicated in human cancer. However, only the ABL tyrosine kinase has a clear causal relationship with the initiation and the progression of human cancer. ABL have an oncogenic version, and this oncogenic form of ABL tyrosine kinase has a clear causal relationship with the initiation and the progression of CML. The normal ABL is P140 CABL. The oncogenic version, which is involved with CML, is P210 bcr-abl, and the retroviral ABL called P160 GAG-abl. Let's talk about each of them. Let's start with P140 CABL, the normal protein. The origin of all the members of the ABL tyrosine kinase family is the normal ABL gene. The size of this protein is 140 kilodalton. It's structurally quite complex. You can find there SH3 domain, which can bind proline-rich regions, SH2 domain that can bind to tyrosine phosphorylated proteins, protein tyrosine kinase, nuclear localization signal, another region which is proline-rich, can bind to SH3 domains. Here you have DNA binding region and actin binding domain. Well, quite complicated protein which have a lot of different functions related to the different domains that appears on this protein. It's highly conserved in the evolution. You can find ABL in nematodes, fruit fly, and of course in mammals. It is expressed in creatures representing different stages of the evolution. This is a suggestion that the normal ABL is a very important protein. So how the ABL is working? In normal cells, the P140 CABL has a very weak tyrosine kinase activity. It can be found in the cytosol with low level of the kinase activity, but the ABL can shuttle between the nucleus and the cytoplasm. And the ability to move from the cytoplasm to the nucleus and vice versa is based on the low level of kinase activity. When this protein is phosphorylated, it cannot move throughout the nuclear pores. The nuclear P140 CABL tyrosine kinase is activated in response to DNA damage. So when the ABL kinase activity is induced by DNA damage, the ABL start to phosphorylate substrates in the nucleus. And here, those substrates are going to activate apoptosis and the cell is dead. So the ABL tyrosine kinase is protecting us from DNA damage. Cells that are experiencing DNA damage are going to be killed by the ABL tyrosine kinase. The P160 gag-ABL. The ABL was discovered in 1983 as part of a genome of a retrovirus that causes the Abelson leukemia in mice. That was the second oncogene that was discovered after SARC by using retroviral viruses as a probe. P160 gag-ABL is the protein that you can find in cells which are infected with those retroviruses. The P160 gag-ABL consists of most of the ABL tyrosine kinase. You're losing the SH3 domain and the first exon before the SH3 domain, but otherwise you have the SH2 domain, the kinase, and everything else. But you also have a gag from the viral genome. The result is 160 kilodalton protein. The amino terminal part is the viral gag protein. The carboxy terminal part was designed ABL after the scientist that was initially found the virus in mice with certain leukemia and lymphomas. The P160 gag-ABL has a strong tyrosine kinase activity, which is essential for the ability of the virus to transform cells. You're making mutations in the kinase domain, which make it kinase dead. The virus cannot transform cells. So the kinase domain is critical for the cancer transformation by P160 gag-ABL. You can find P210 bcr-abl in 95% of patients which carry an abnormal chromosome called the Philadelphia chromosome in the CML cancer cells. One of the products of the Philadelphia chromosome is an abnormal protein with strong tyrosine kinase activity. The amino terminal part is the BCR protein, and the carboxyl terminal part is the ABL tyrosine kinase. Here you can find SH3, SH2, the kinase, and all other elements that you can find in the normal ABL. The size of this protein is 210 kilodalton, and that's why we call this protein P210 bcr-abl. So what is this Philadelphia chromosome? The Philadelphia chromosome is a genetic abnormality caused by a reciprocal translocation of genetic material between chromosome 9 and 22. This is a human karyotype, chromosome 9, chromosome 22. Chromosome 22 is the smallest chromosomes that we have in our genome. And the chronic myelogenous leukemia is caused by reciprocal translocations between the ABL gene on chromosome 9 and the BCR gene on chromosome 22. This reciprocal translocation is putting the ABL tyrosine kinase gene in a proximity to a gene called BCR. The fusion is involved with the protein which is in frame, and the protein consists of both BCR and ABL. And this is a protein with a strong signaling ability, making white blood cells to proliferate in more significant way. Normal cells versus CML cells. Normal cells have their chromosome 9 with the ABL gene, chromosome 22 with BCR gene, and upon the reciprocal translocation, the ABL gene is going to be fused with the BCR gene. So what we are going to have is Philadelphia chromosome, which is most of chromosome 22 plus the end of chromosome 9, which consists of ABL, while the chromosome 9 is going to have the small portion which comes from chromosome 22, but there is no ABL on chromosome 9 after the reciprocal translocation. There are two types of chromosomal fusions that can take place between the BCR and ABL. In both, exons 2 and 11 of ABL are involved, and exon number 1, which is a short exon, is usually not there. But in P210 bcr-abl, the exons from BCR are 1, 2, and 3. So you have, after all, a longer protein as compared to the other option. P185 bcr-abl, here the fusion is in the intron between exon 2 and 3 of the BCR. So the BCR exons that is involved with the fusion here in P85 bcr-abl is exons 1 and 2 of the BCR with exons 2 and 11 of c-abl gene. I will concentrate here on P210 bcr-abl, which is involved in most of the cases of CML. P185 bcr-abl is involved in another childhood leukemia called acute lymphocytic leukemia, and we are not going to discuss this. So let's see what's happened when we have the normal ABL that can cause apoptosis upon DNA damage versus leukemia cells here. So we have the normal ABL, the normal BCR, both of them are cytoplasmic protein. The ABL can shuttle between the nucleus and the cytoplasm, and the ability of ABL to move from the cytosol to the nucleus is dependent on low-level kinase activity. Because when ABL is active, it's not going to enter the nucleus. In bcr-abl, the ABL is active all the time. There is high level of kinase activity. And as the result of this, the ABL cannot enter the nucleus. The P210 bcr-abl has a constitutively active tyrosine kinase, and P210 bcr-abl is retained in the cytoplasm. It is kinase dependent. The nuclear localization signal is not available when the ABL is active. The P210 bcr-abl by itself have several activities which are quite different from what we have from the normal ABL. P210 bcr-abl protect hematopoietic cells from apoptosis and activate downstream signaling pathways. bcr-abl act like a normal tyrosine kinase that when it is active, it's going to activate bunch of different pathways. First, GREPSOS, RAS, MAP kinase is going to be activated. It's activating also JAK-STAT pathway. It's also activating other pathways, which each of them is involved with separate activity related to cancer. So the transforming effect of bcr-abl is mediated by several downstream signaling pathways, normally activated by receptor protein tyrosine kinases, including the RAS, RAF, MAP kinase, JAK-STAT, PI3 kinase pathway, FAK pathway. All of those pathways are involved with different activities that can satisfy, after all, the hallmark of cancer. So P210 bcr-abl is a dominant oncogene because it can activate several activities which eventually are going to be essential for the oncogenic transformation. Well, all you need is one hit. All you need is bcr-abl in order to give the cell most of the hallmark of cancer that can satisfy the transformation of those cells to full malignancy. And that's make the bcr-abl as a dominant oncogene. When there is bcr-abl, there is CML. When bcr-abl is not there, usually there is no CML. During the 80s, we start to understand that bcr-abl have a clear causal relationship with the initiation and the progression of cancer. At that time, the 80s, we had some information that was quite clearly indicating the P210 bcr-abl and the Philadelphia chromosome as the oncogene which is involved with a specific unique cancer. One, Philadelphia chromosomes appears in more than 95% of CML patients from the initial diagnosis stage of the leukemia. So when you find a person with Philadelphia chromosome, this person is by definition have CML. And when you find a person which was diagnosed with CML by other ways, this person is very likely to have the Philadelphia chromosome, which is expressed to make the P210 bcr-abl. The relationship is straightforward in most of those cases. Later on, that was something that came afterwards, the early 90s. The expression of P210 bcr-abl in mice induced leukemia in most of the treated animals. There is another thing, that the appearance of the second Philadelphia chromosome is an indication for the progression of the disease to the accelerated and blast crisis stage. I want to take you back to the end of the 20th century. Physicians were dealing with clinical oncology, like chronic myelogenous leukemia. Patients came to see the doctor, which gave them certain treatments that were available at that time. However, in parallel, we were able to understand more and more the molecular genetic of cancer. Totally two different directions that were not talking with each other. During the end of the 20th century, the molecular knowledge of CML had no effect of the way that we can treat the disease. Basic scientists and physicians had pretty much nothing to say to each other. The treatment of CML had nothing to do with the molecular abnormality that causes the disease. And the treatment had poor outcome. Most patients with CML ended up dead from the disease or from the treatment itself. One treatment which was available at that time, which happened to result in cure in some cases, was based on the fact that CML is a stem cell malignancy. CML is a problem of the bone marrow. When you are checking the bone marrow cells, you can find the Philadelphia chromosome in the stem cell compartment. So the idea was to treat the disease by killing all the hematopoietic stem cells. If you kill all the hematopoietic stem cells, you are killing also the cells which are involved with cancer, the Philadelphia chromosome that happen to be the majority of the cells in this stage. And the treatment called bone marrow transplantation. Bone marrow transplantation is a special therapy for patients with cancer or other diseases which affect the bone marrow. The goal of bone marrow transplant is to replace the bone marrow cells and to restore its normal function after a high dose of chemotherapy or radiation, which are given to treat the malignancy. A bone marrow transplant involves taking cells that are normally found in bone marrow to replace the bone marrow. I mean, people must have their bone marrow cells in order to have blood cells. The immune system, the red blood cells, they are all originated from bone marrow. If you're killing the bone marrow cells which are involved in a certain disease, you need to replace them with healthy cells. So the goal of the bone marrow transplantation is to transfuse healthy bone marrow cells into patients after their own unhealthy bone marrow cells have been eliminated. So how this process is working? First, we need to find a matched donor. Healthy person which happen to have a similar HLA type on their immune system cell. Again, everybody have his own markers and it's quite difficult to find that matched donor, but it can be done. So first, you need to find the donor before starting this process. And when you have the donor cells available, you can start the procedure. The recipient, the sick person, is going to get a very significant radiation chemotherapy that is likely to kill all the bone marrow cells in their body. Very aggressive treatment, radiation chemotherapy, which makes this person to lose all the cells in the bone marrow. Now the process is to infuse the normal cells to this person that now do not have its own bone marrow cells. The bone marrow cells are likely to start to grow in the bone marrow, and eventually they can start to differentiate, produce all the different cells that we need in the blood. So bone marrow transplantation in CML. There are two types of bone marrow transplantation. autologous bone marrow transplantation and allogeneic bone marrow transplantation. If you are lucky enough and for some reason you kept some bone marrow cells of your own before you had the disease, you can use them. And today we are talking about umbilical cord blood, which can be collected from a newborn and can be stored in liquid nitrogens and then can be used if the person need bone marrow cells. That's one option. Another option is to collect the patient's own stem cells and to eliminate the cancer cells from the bone marrow and then reinfusing them after the patient has received high dose of chemotherapy or radiation therapy. And that's another option which comes with the risk that you are not going to clear all the cancer cells and you don't need a lot in order to bring the disease back. Allogeneic transplantation requires tissue type HLA, matched donor, who can be related or unrelated to the patient. If you're lucky enough to have a twin brother or sister, you can get allogeneic transplantation from them, which is likely to have quite similar HLA antigens. The donor is going to be matched donor in most of the cases, or you can go further, brother, sister, even parents. And actually today you can take it into another direction in order to find matched donor, because you can find the matched donor in the databases. Today the HLA type is recorded, and we have a lot of situations where people which do not have a matched donor from their family, the physicians can find the matched donor by searching the database around the globe. Now, with both cases, they may result in cure. You're killing the bone marrow cells, the cancer cells, and then the patient can start from fresh by the bone marrow cells that do not have the Philadelphia chromosome. However, there are problems that can be found in both. Autologous bone marrow transplantation are coming with the risk that the cancer will return. Clearing the cancer cells from the bone marrow is limited. This process is not totally efficient, and you can find some situations that the autologous bone marrow transplantation ended up where the disease came back. But let's concentrate on allogeneic bone marrow transplantation. Here we have donor limitations. I mean, there are a few people that have enough family members, brothers, sisters are different, and when it comes to parent, child, or child-parent donation of stem cell, they are related only in 50% of their genes. So that's quite complicated situation. So donor limitation is always a problem in bone marrow transplantation. It takes time to find the matched donor, even today when you can search the database and try to find a matched donor by using database on computers. And when the donor is not matched appropriately, there is a chance for graft versus host disease. Graft versus host disease, GVHD, is the problem that the graft, which is the bone marrow, is attacking the host. The immune system, which are made by the bone marrow, which are coming from someone else, are going to see the body of the host as non-self. That's a problem because you're going to have a lot of immune cell activity that are going to affect many different tissues. In this condition, the recipient new immune system created by the stem cells provided by the donor attack the recipient's body. Another thing is, during the time that you are waiting that the immune system will be restored, you are more susceptible to many infections. Infections which are coming from the outside world, but even infections which are inside you to start with. We are all infected with viruses, like herpes viruses, and viruses like CMV, like EBV, can take advantage that the immune system is not there, and they can start to cause problems which are very difficult to treat. During the time that they are waiting that the donor cells will be established, they are highly immunocompromised, can be infected with pathogens that normally are not causing major problems when your immune system is there. And still, there is high percentage of relapses, means that the tumor is coming back, especially in autologous bone marrow transplantation, but not only. So even if you think that you cleared all the bone marrow cells from the body by radiation and chemotherapy, that's not happening all the time. 2001, up until that time, the only option for cure was bone marrow transplantation. So what is the outcome of bone marrow transplantation? This is survival. This is years after the transplant. If we will concentrate here 1 year after the bone marrow transplantation, we can see that the best scenario is when the patient is going through the bone marrow transplantation during the first chronic phase. But still, you are losing almost 50% of the patients who are killed mainly from the procedure. When you are waiting a bit longer to the accelerated phase, 1 year, well, 60% of the patients are going to die. If you are waiting to the blast crisis, very few are going to stay alive after the bone marrow transplantation. So bone marrow transplantation can result with a cure, but most of the patients are not going to make it. That's not good. And here we have a problem. Well, when we are talking about the chronic phase, you're talking about 4 to 6 years of chronic stabilization. In all cases, the untreated chronic phase is a time bomb. In all those cases, the patient is going to progress to the accelerated phase, which will follow by blast crisis phase. What it means is that if you're not using any treatment, you're going to have for quite a while that no treatment is giving you a better outcome as compared to people who were treated, who had the bone marrow transplantation. So the decision to do the bone marrow transplantation or not was extremely tough. I want to take you to the end of the 80s. I was a young PhD student in Hadassah Medical School in Jerusalem, working on the bcr-abl. We were in the same campus together with Hadassah Hospital, and we were collaborating from time to time with physicians from this hospital. One day I was asked to teach a physician from the hospital a certain scientific protocol that I was using in my studies. So she came to the lab and I started to do my experiments when she was there. This physician was expert in chronic myelogenous leukemia. She had patients at different stages of the disease, and they were calling her all the time. At that time, we didn't have cellular phones, and the way to be in contact with your physician is by calling the center of the hospital, and the center was sending a message on the beeper that made the physician to call him immediately to the center to have the discussion with the patient. The only telephone that we had in the room is the one that was on my bench. And when she was talking with the patients, well, I was there. And I remember two cases. One of them is 50 years old patient that was diagnosed in the accelerated phase. And the physician told him that he should take the bone marrow transplantation. That's the time to do it. And the patient said, well, my daughter is getting married in 2 months and I want to be there for her. So the physician, together with the patient, made the decision to wait with the bone marrow transplantation. 2 months later, when he was back, he was in the blast crisis stage. Nothing can be done in the blast crisis stage. Here, the bone marrow transplantation on time may save the person, maybe. We don't know. And I remember another person, again I'm going to use just the first name, Ofer. Ofer did PhD in Weizmann Institute. And he was diagnosed with a chronic phase at that time. And the physician wanted him to do the bone marrow transplantation now. Ofer said, well, I just started my PhD in Weizmann Institute, and that's my dream, and I'm not going to be interfered by the chronic phase of CML. I want to finish my PhD first, and then we will talk. So that was the discussion that they had over the phone next to me in my room. I haven't heard from Ofer for quite a while. But in 2007, I went to visit the Weizmann Institute website, and I found a message that Professor Ofer such and such from the department such and such was killed from cancer a week earlier. Yes, Ofer was able to complete his PhD, to do postdoctorate fellowship, to come back to Weizmann Institute, to become a professor over there. And he died like 70 years after the time that he refused to have the bone marrow transplantation. So that can tell you what a problem we had at that time to deal with chronic myelogenous leukemia, where no treatment in many cases was the best scenario, at least for a short term. Yes, we know that CML is quite unique. CML is a disease that we know what is the oncogene which is involved with the disease. It's more or less 1-to-1. P210 bcr-abl is CML and CML is P210 bcr-abl. I mean, it's different as compared to other cancers at that time. And again, when we were looking into the way that we were doing clinical work on patients with CML versus the molecular knowledge that we had at that time, well, physicians that were doing the clinical work had nothing to say to the scientists that are studying the molecular genetics of cancer and vice versa. The information we had from the molecular genetics of cancer had nothing to do with the treatment that was offered to the patient. Bone marrow transplantation is not about P210 bcr-abl. It is not affecting specifically cells with Philadelphia chromosome. Bone marrow transplantation is used for a lot of other things, including different genetic diseases or other type of cancers. So the molecular defect that we have in CML was not used at the time. And the question was, can we put the molecular geneticists, the basic scientists, and the clinicians together? Can we use the information in a bit more productive way, that the clinical oncology will get some benefit from the knowledge that we have from the molecular genetics of CML in this case? So the question that we asked, does the understanding of the genetic abnormality of CML may allow to deliver genotype specific therapy? And that will be the topic that I will discuss with you in the following lecture.